So good morning, everyone. Let me share my screen. Can everyone see the screen well? Yeah, my name is Jummin Peng. I'm a faculty member at St. Jude Children's Research Hospital. Actually, I'm in the Department of Structural Biology, also the Developmental Neuron Biology. In addition, I'm a director of the Center for Proteomics and Metabolomics in the hospital. So I would like to really thank Jeff for the invitation to this meeting. This is my first time to attend this meeting. And then I actually received an email from Jeff a few, maybe 1 or 2 years ago, and then get to know the field about CHAMP1 disorder. And then at the hospital, a research in the hospital, we actually feel like whether we can do something for this disease. So first, I would like to give you a very brief introduction about St. Jude. You can see St. Jude has been a children's hospital largely focused on pediatric cancer research and cancer treatment in the last more than 50 years. So you can see this is the hospital, and then it's painted pink, right, for children's, and then our research building is right around here. So this is at night. We are in Memphis. Some people may ask us why we're in Memphis and why called Memphis. The city is named after the ancient city Memphis along the Nile River. We are close to Mississippi River, so you can see the river bridge here, and then we also have a pyramid here, and then our hospital is right here. So although initially St. Jude has been focused on pediatric cancer research, in recent years, the hospital is also looking for expand our scope to understand and treat pediatric neurological disorders. So you can see here we have a program called the Pediatric Translational Neuroscience Initiative, and then has been led by our CSO, Dr. J. Paul Taylor, and then with several other leading scientists like Dr. Peter McKinnon, and the physician scientist Richard Finkel, and then also we have an organizer called Kristen. I believe Jeff had been in contact and talked with Kristen previously. And also myself has been discussed with them about this project, and also they are very supportive, and then they would like to see how the project can go, and in the future potentially will be supported somehow by the St. Jude. So I have to say I'm new to this field, although my research is in neuroscience and also molecular diagnosis and study of the human diseases. This disease is still very new to me, and then I haven't learned a lot. I watched some of the videos and also hear Dr. Talaka's talk about a mouse model, and then here basically you can see a copy from your wonderful website. And then I will skip here and then talk about what my current understanding of this disease. And then you can see the majority of the disease, you can see is caused by a mutation in a specific gene called CHAMP1. And then the mutation is largely called either missense or microdeletion or frame shift or nonsense mutation. All those mutations can cause the disappearance of the protein. Right? So this protein, since a human has two chromosomes, two copies of the gene, and then if you have one mutated, you probably have a less amount of this protein, and which can potentially leads to the occurrence of the disease. So there are some early studies, and then to show the protein is located to chromosome and they involve the cell cycle. And then I would like to just skip that. And then I think that has been discussed by other previous speakers. And also the interactome study of this protein also shows many of the protein interact with CHAMP1 are also involved in chromosome activity. And then also largely the mutation of those genes is linked to development disorders, which has a certain similarity to the CHAMP1 symptoms. Okay, so let's talk about what really we would like to do. So we actually have been using the inducible pluripotent stem cell to model the disease. So this is a relatively new technology, and then it's a Nobel-winning technology. The two scientists initially involved in this study, especially Dr. Yamanaka, they won Nobel Prize in 2012. So in this year, so the basic idea is you can take any cell, largely a skin cell, the fibroblast cell from anyone, and then you can reprogram the cell and then allow the cell can go back to the stage and like the close to the very early stage of the cell, like fertilized egg, but it's not really there, but it's close. And then in this process, and then this cell has the potential to develop into any other cells, and also even develop into in organism. I believe in the mice, they can already reprogram it, and then the cell can actually give a new birth of a new mice. But of course, this is prohibited in human. In this process, you can see you can either take the healthy individual or patient's individual skin cell, reprogram, use this cocktail initially invented by Dr. Yamanaka. This is four transcription factors, 4 factors. And then after that, you can generate we call it IPS cells. And then those cells can further induced, and then you can generate a neuron from here, and or astrocytes or microglia. Those are different brain cells. And then in addition to that, you potentially also can generate an organoid. We call it organoid. It's like an organ, but it's not a really an organ. And then you can see multiple brain cells and grow in a petri dish, and which is relatively small. You can see by naked eye, but it's not very big. It's not a really an organ, so that's why we call it organoid. So to design this experiment, we think if we can take those cells, can we compare the healthy individual with the patient's cell, right? So in this process, we can take the human iPS cell from the health individual and also from patients. So for the healthy individual, potentially we can introduce a gene mutation, right? And then if the mutation is introduced, which potentially will cause the disease, and then can generate this is a specific cell which are associated with disease, potentially can mimic the condition in the disease. And then on the other side, we can also take the patient cell at the beginning as a startup, And then we can use the technology, we can correct this mutation to generate a Y type cell. And then we can also compare the corrected and with an uncorrected cell in a Petri dish. Why we want to do that? Because when we do the correction, we can generate what we call the isogenic cell. That means this cell and this cell are largely similar. Potentially all of them are similar except this specific gene. But however, we know the technology itself can generate some byproducts, or this is not 100%, only focus on one gene, but it's actually close enough. So that's why an isogenic analysis is important. Why this is important? Because every healthy individual and every patient are quite different, right? So for example, if you simply compare my gene with your gene, you will find many differences, although both of us are healthy at this stage, right? So in these cases, if we use isogenic comparison, which can largely allow us to reach a conclusion. So the gene editing is also a Nobel's winning prize technology. Basically, two scientists won the Nobel Prize in 2020 for discovering the CRISPR-based mutation correction technology. So you can see those are very novel technology, right? Only actually available recently. So you can see here, we can obtain the patient IP cells, and then use this specific technology. I'm not going to talk about the detail to correct the mutation, right? And then go to this one. And then we can develop the brain organoid. And then one is from the patient, right? Another one is from the corrected one. And then we can, once we have this brain organoid, we can do all the follow-up analysis. We can analyze right, DNA, RNA, protein. We can even do this fancy single cell RNA-seq analysis to analyze all those cell types. I believe this is important because we believe the mutation of CHAMP1 may not affect every cell. They only affect the small sets of the cell, and then some cell development is more sensitive to the dosage of CHAMP1. So we believe the cell type analysis is very important. So that's what we are applying to do. And then after we understand the defects caused by CHAMP1 mutation, which allow us to think about what's the best strategy to treat and then to correct those the consequence of this mutation. So you can also probably imagine the CRISPR itself can be used as a gene therapy, right, method to correct for the mutation in patients. I believe this is a very, very active field. And also I believe Jeff is also actively talking about with experts in this field to see whether it's possible to use the CRISPR technology to correct the mutation directly in patients. I believe some of them have been already in clinical trial in different diseases. For example, like sickle cell, right, and the diseases and other diseases as well. This, I have to say, this is a very active research field, and then has a high potential in the future. Right. So what's our own progress? I think when we first talk about this project, we really think about what to do, and then we waited a while to really go through all the legal and then materials transfer process. We obtained first we obtained one of the iPS cell. You can see here called CHAMP1. The gene, the coding gene is 1489 C, actually mutated to T, and then which cause a protein. Initially is an arginine, right, and an amino acid to a stop codon. So this one actually caused a serious. This is a very strong mutation, can cause the loss of this protein. And then after CRISPR correction, we already generated the potential wild type version. You can see you can T originally now change it back to C, right? From here, initially in the patients is C to T, but now we can change back from T to C. And then we already obtained 2 clones in our lab. I think this takes almost several months to do. And then you can see these 2 clones, right? And both of them actually are correct. And then also we performed another correction, use a different line. And then in this case is on a different position. Is a premature stop codon at position 5 on this new open reading frame, because this is a deletion right here. So you can see on the right-hand side, the CT, right, CT sequence are missing, right? And then, so that's the original mutation. And then after correction, we inserted the CT back, the 2 nucleotide back. We also got 2 clones right here. And then we actually now have basically 4 clones, and then from 2 initial patient samples. So we are in a process to differentiate and then to generate the cerebral organoid in process. So the process is like this. We take this cell, we also can maintain this cell in a petri dish. And then at a certain date, we will use a cocktail to induce the formation of an embryonic body. We call it EB. You can see it looks like that. And then after that, we began to induce them. And then all the way, eventually we are looking for to form this, we call it organoid. And then they have a layer of structure, and then which contain many type of neuron cell and also astrocytes. And then once we have that, we're going to analyze this organoid carefully by analyzing the RNA and then protein and also the single cell type in those organoids. So that's our goal. So I will stop here. I think I will have to say this is a very pioneer design, and then we actually are moving along this direction. Probably next meeting we should have results from the organoid we obtained. Yeah, thank you very much. And then I would like to take any questions.
Thank you so much, Dr. Peng. Can you hear me okay?
Yes, yes.
Okay, wonderful. That was amazing. I want to open it up to for a few minutes before we go to lunch for any questions from anybody. Anybody have any questions? Hang on one second, Dr. Peng.
Okay.
So I may have missed it, but you're making organoids from just one mutation, or you're going to make a lot of different mutations and compare the organoids?
Yeah, we make the organoid from two mutations.
Once you get those phenotyped, are there plans to add any other mutations?
I think we would like to see to get the initial result and then to see what really happens. I believe all the mutations currently cause this loss of the protein, right? And then this is not a structure change. Basically, the protein is can be probably represented half in the mutants. We will see what we can really get from here, and then we can design what's the next step.
I'm getting the impression, I certainly don't know as much about this as most people in this room, but I'm getting the impression that there are some mutations that change the structural conformation and are not necessarily haploinsufficient. So it could be really interesting.
Right. If yeah, I think from a literature currently, I know is all somehow cause the haplo deficiency. So yeah, definitely, as I said at the beginning, this is new to me. If there are some actually cause structural changes, we will be glad to take additional mutation and then to correct those as well. Yeah.
As an update, I know that there are several mutations that should be available maybe Q1 of next year from Simon Searchlight. I think we talked about that. So I'll get you an update as soon as we have one as well.